Folks in Zoom, it looks like y'all were not able to hear because of some technical issues. For the folks in the room, I'm going to trace back, do a very quick overview so that folks in the Zoom can understand what I'm talking about a little bit more. So apologies for the delay here. So to catch up people in Zoom, I'm going to just briefly introduce NF-Core, scnanoseq, but then I'm going to talk a little bit about NF-Core and tell you all about like the past of the history of this particular workflow as well as the future. So very quickly, I'm not going to go into as much detail for the sake of time. This particular workflow is a workflow for secondary analysis of 10x genomic single-cell, single-nuclei RNA-Seq data, specifically targeted for the Oxford Nanopore sequencing instrument. So this is for long-read RNA. I'm going to kind of touch on this as I walk you all through the diagram, but this was released in 2024. We've had a couple of patches since then, but we actually published the paper September of last year. And this was developed by the UAB Biological Data Science Core. So I was actually right here, right at the middle of explaining this diagram. So I'll kind of do a quick trace back. As I've said, this is a secondary analysis workflow, so that means we're starting with SQ files. It would be essentially the equivalent to, say, Cell Ranger if you've ever worked with short read data, but this is specifically targeted for long read data. And when you look into the details of this diagram, you'll see that there are a couple of QC steps that are repeated at different time points. And the reason for that is that we wanted to give you to the user the ability of having them review how many reads were dropped after each one of those steps. With that being said, these QC steps can be skipped if you want, and we provide different options on purpose because some of these tools will perform well with long-read data, but some won't. So you don't need to do all three, you can just choose one. The very first read trimming or the read processing step will be with just quality filtering with a tool called NanoFilter, because this is still data that can suffer from quality, as I've discussed earlier. And then the next step will be to do barcode detection. This, again, is single cell 10x genomics data. So the libraries will contain cell barcodes. And to quantify it appropriately, you need to be able to detect it as well as extract it. Detection is done with Blaze, extraction is done through a custom script. After that, we then go ahead and we start the alignment steps as well as barcode correction. And this is actually kind of where I was before we got interrupted with the Zoom issues. What I wanted to highlight here is that the workflow provides two ways of quantifying the data. One is through the quantification to the genome, and the other is through the quantification to the transcriptome. So I was making a little bit of a comparison for those of you who may be familiar with NF-Core RNA-Seq. They do the same thing there where they provide, for instance, a way to align it to star, as well as to pseudo-map or quasi-map to Salmon or Callisto, where you use the transcriptome. So if you do want to choose one method versus the other, you can do so, but you can also just choose both, and it's up to you. With that being said, of course, at the alignment step, that will be a point in the analysis that has to be done separately because we need to pick the right references. But while the alignment is happening, we also go ahead and we perform barcode extraction, which at this time it is currently being performed with a custom script in the pipeline. For those of you who may not have realized, it's very common in secondary analysis of 10x data, because even though we do have a cell barcode list that always gets checked against a whitelist, there's a chance that some specific nucleotides in those cell barcodes are incorrect, and that may be due to sequencing errors, things like that. So the workflow does allow you to correct for that as long as it is within a small probability that it is truly fully incorrect. So we allow for that to a degree. After the alignment is done, we then collect the alignment metrics just to provide that to the user, and this is actually compiled into the multi-QC report. And then importantly, we go ahead and we add the metrics from the cell barcodes, including corrected metrics, back into the BAM files through a process called barcode tagging. So this is pretty important if you want to ever look into detail, like the BAM files allows you to check how many reads contain cells that were not correct, cell barcodes that did not need correction, cell barcodes that were corrected. And there's a specific set of columns in the BAM file where you can extract that data. So that is all present after this particular step. After barcode tagging is done, we then move forward with deduplication. So deduplication is a step that can be turned off by you if you like, but this will only be a possibility if you choose the isoquant path or the genome quantification path. And this is a flexibility that isoquant provided to the users, However, we do turn that on by default. I do think that deduplication is the best approach is better for you, but if there's a reason to skip it, you can. With that being said, ORFISH does require deduplication, so that will always be turned on by default. And this is done with Picard tools or Yumi tools. And then we move forward with quantifying your transcripts, right? What are the isoforms present? And this is where these two tools that I've kind of talked a little bit about come into play. We've got currently isocoin into the pipeline, as well as ORFISH. One key distinguishing factors between the outputs that you get between these two is that isocoin will provide you, in addition to the isoform matrices, gene level matrices as well. ORFISH currently only provides transcript level matrices out of the box, although we are looking into standardizing that process in the workflow in the future. But you can do that yourself, but we would really like to work with tool developers such as Michael Love to put that functionality into packages such as TX Import and TX IMETA, because this is something that we really wanted to target it in here. We wanted to standardize these types of process. But you can do that. It's not possible. I'm happy to chat with you with ORFISH if need be. And then at the end, we perform one last step, one more quality control step. And it is done with Sura, but it doesn't go into depth. The only goal of this particular step is to provide you with some key metrics that we believe is very important to secondary analysis. So kind of going back to Cell Ranger, for those of you who've ever run that pipeline, if you open a report, you will see some very important metrics right at the top. So things like how many cells, right, were detected, how many features were detected, as well as how many reads per feature. So this is essentially what this is doing. We combine Sura as well as the metrics from SAM tools to compile a custom table that we output into the multi-QC report. We also do have some high-level plots, but that doesn't go into the multi-QC report, but it is present in the results. And then we deliver the data, which are the key outputs will be those matrices that you can put into downstream tools such as Sura or SquidPy, and as well as the multi-QC report, which will compile most of this analysis. So that's the overview of the workflow and where it is at the moment. We'll be touching a little bit in a few slides about where we started and then again, where we'll go. So with that being said, though, before I talk more about scnanoseq, I wanted to kind of digress and tell you about NF-Core, in case you're not familiar with this particular community. NF-Core is a community-led effort to curate, develop, and standardize Nextflow workflows. This is actually an international community, and the great thing about them is that they have a lot of different workflows. These are all freely available. Any community member can use it, and any community member can also contribute. A big reason as to why NF-Core has been so successful has been its open source community. So if you look at the latest metrics from NF-Core, you can see that there's been 87 workflows that have been released, 44 are under development, and only 13 got archived. So these are workflows that are not under maintenance anymore. So I think you can really easily appreciate that this has been a very successful community when it comes to like the development of standardized workflows. But importantly, beyond, right, these workflows that are being developed, NF-Core goes beyond that. They're not just looking to develop workflows and that's it. NF-Core is about the community. What they have really done by working with community members is to really standardize and really ensure that fair practices, reproducible practices, scalable practices were in place for these types of workflows. So that includes not only standardized coding, as well as standardized documentation, right? There's nothing worse than finding a great tool that is not well documented. Standardized pipelines in the context of versioning, so versioning control workflows really well, and community outreach too. So they've done really good, good job at involving different people across the globe to really not just teach, they have training material, but also involve them in opportunities to refine their skills, such as hackathons, things of that nature. So this is what NF-Core is about. It's really this international community. And one thing I want to say about NF-Core is that all NF-Core pipelines will be developed using Nextflow. But not all Nextflow pipelines will be NF-Core pipelines, because to be an NF-Core pipeline, you do need to follow the NF-Core practices. It doesn't mean that non-NF-Core pipelines are not good. There are plenty of really good examples of very good Nextflow workflows that are not part of the NF-Core community, but that is one distinguishing component there. You can make a Nextflow pipeline however you wish. But to be part of an F-Core, you need to involve an F-Core early on so that those requirements are clear. Reproducibility in particular. I wanted to touch on this because this is a very important component of all NF4 workflows. They've put a lot in place to ensure that these analyses would be reproducible. And this is just a few examples. It's not all of them, but one of them is, of course, using Xflow as a starting point, but also ensure that the workflows themselves or every single process would be reproducible by the use of containers. Because even though there is native support to containers in Xflow, you don't need to enable that. You could just run an Xflow that doesn't use containers or doesn't use a container environment, but NF Core enforces, so to speak, that every single pipeline should use containers. Version control, every single source code must be hosted in GitHub and it must be version control. And on the source code side, they actually have specific templates that a developer should use. They recommend users or developers to use centralized modules. So modules or centralized modules are modules for standard tools such as like Star, Minimap, SAMtools that have already been reviewed and actually sit in a common repository so that multiple people can work on it and you're just using that central module instead of reinventing the wheel. Now, that's not always going to be feasible. We actually, in our own workflow, had to create our own modules because not every single module for workflows will exist in an F-Core. But if you do create one, you still need to follow the best practices on how to do it in the context of applying syntax, the syntax rules that is recommended by NF Core, so on and so forth. And even when you're done working on your workflow, let's say everything is working, you still want to go ahead and perform linting, right? So linting will ensure that the actual syntax across your workflow is indeed following what they recommend as best practices. And that's just to make code maintenance a little bit easier. Because if you have 87 pipelines, right? We have 87 pipelines today published as part of NF Core. Imagine if they were all different. Imagine if they were all written different. It would be a nightmare to maintain them, and it would be a nightmare to also even understand them. So even though I'm not the developer of, say, NF Core ATAC-seq, I can go in there and understand that workflow really easily because they follow a standardized format. So that goes a long way. And then lastly, of course, in terms of releases, these are, of course, done at a particular time points during the development of a workflow. And if you do want to release a pipeline as part of the NF Core community, you do need to get it reviewed by community members. So it's always going to be 2 members. You can have members of your own team review it if they're members of NF Core, but usually it's good to at least have someone external review that as well. So again, that is a lot to NF Core, but one thing I also want to emphasize about them is that they have really captured a high level of community engagement. And that again includes not only activities such as hackathons and training, but they have a really active presence online. So if you're interested in joining, here are some of like the channels that they are active in. Slack is great. There is a lot to the Slack channel in NF Core, I'll warn you all, but it's just a matter of learning how to like maintain that, like turn off or turn on specific notifications. It's a great resource overall. And importantly, if you've never really seen the NF Core website, I wanted to do a quick walkthrough with it and give you an example documentation page from our workflow. So if you go to the NF Core website, there is a lot there, but the key thing will be the pipelines. And if you do click on View Pipelines, you're going to encounter a page similar to the one that I have here below. And this is where you're going to be able to browse the workflows. And you can actually start typing specific keywords such as like RNA-Seq or single-cell RNA-Seq or ATAC-seq, whatever, and it will filter that down a little bit for you. The ones that are at the top will always be the ones that just had a release, so that always gets updated according to releases. In terms of documentation page, no matter what pipeline you use, the documentation page will look very similar to this. So this is the one for NF Core scnanoseq, since this is what we're discussing today. So we'll walk you through this very quickly, but understanding that this is one of those things that NF Core standardized is their documentation, right? So at the top, we're always going to find the pipeline name. Then we're going to find the specific version that is the latest. So our latest version right now is 1.2.1. We're about to release a quick patch. So probably later this week will be a 1.2.2. And then we have the link to the source code. Importantly, this documentation page is also updated, has the pipeline evolved. So if you're using, say, I'm going to say 1.0.0, our documentation page will have some differences. So it's important to go back to that specific version if you're using that version to ensure that you're using appropriately, and you can change that on that particular tab that I've highlighted. And then in terms of like information about the pipeline, there are different tabs that you can access to find out more. And that includes the introduction, the usage, so how you use the pipeline, obviously, the parameters, the outputs, the results from what we call the test profile that are present in AWS, as well as details about the releases. And just because this is like the default page that you encounter, you're always going to first find the introduction right at the top, as well as the meta diagram right there that's below. I already presented that earlier, so I just cut that off for the sake of space. And then last but not least, one last note about NF-Core. Even though NF-Core is heavily focused on the standardization of Nextflow workflows, again, they do a lot outside of that. They do training, once again, they do hackathons, and they also maintain these repositories that really help you use Nextflow workflows as part of your day-to-day work. So this is an example that I picked because it's relevant to us here at UAB. This is the NF-Core configs repository or the page about it. And what this provides is that it provides you with a series of Nextflow configuration files that allow you to quickly implement these workflows in institutional clusters. So Austin and I actually created the one for UAB. We have the CHIA profile in there, and we encourage you all to use it. And if you needed to modify, by all means, start from the source code, the source config that we've provided, and make any modifications that you want. But this enables you to use executors, in our case, it's Slurm, efficiently, so that you're leveraging parallelization of these workflows in a cluster setting. And they have this for multiple institutions across the country. They have this for like cloud services, so on and so forth. So that's what NF-Core is about. And this is why we really value this particular community a lot. And we wanted to contribute to it directly by actually developing one ourselves. That being said, we didn't start there. So this is where I'm going to kind of take a step back into history. And the truth of the matter is that we've actually been working with long-read single-cell data since around like 2021, so almost 5 years ago. Now, this is very initial phase. For those of you who know anything about long-read technology, you will know that long-read technologies will always kind of be a few years behind short-read. So single-cell RNA-Seq really blossomed with 10x genomics around 2018 or so, but most of it was just short read. So in 2021, 2021 was the year that individuals started to like kind of start to use it more rapidly, because in this time frame, Oxford Nanopore had actually released a new chemistry that really improved their quality scores a lot. Because one thing that long-read technologies typically suffer from is quality. But at this time, they really released a new technology that allows us to have at least Q20 plus Fred quality scores in their single cells in any type of sequencing. Now, that doesn't mean you always get that quality score. You still get some that are below. I've seen this myself. So you would want to filter anything that you see fit. But that was a lot more promising for single cell. Up until this point, the only way that researchers did single-cell analysis with long-read technology was to do single-cell analysis with paired, right, long-read paired with short-read, because they needed the short-read data to essentially validate their long-read data and actually help correct that. But as you can imagine, that's very expensive. That can add up very quickly. And on the data analysis side, that's processed very differently. So when we're talking about, technology that can sequence single-cell RNA-Seq without needing short-read data, there were essentially like the methods that were available to analyze that data were extremely limited at this time. And that's when we started to kind of think about this. There were no workflows at that time that were available. So this was, of course, pre-scnanoseq. This was pre-WF single cell. So WF single cell is today the pipeline that Oxford and Nanopore has developed for secondary analysis. That did not exist back then either. They started creating that, I think it was around 2022, which was they ported that over from an older SnakeMake version. There were no other workflows as well. So things like Skywalker, for those of you who are familiar with that particular pipeline, that just didn't exist. But in addition to the lack of workflows, there was also a lack of methods available to process that data. So methods which we now use today, such as Blaze, which performs barcode detection, as well as isoform quantification, so isoquant or FISH. Those methods may have existed at that time, but they were only for bulk RNA-seq data. They were not, they did not enable you to implement them towards single cell. That was also not existent. So we took upon ourselves to kind of create this so that we could support investigators here at UAB on at least on pilot studies, and ideally help them get their grants too. Because our hope was that we could actually work with the UAB community so that individuals could get their grants, and then we could continue to work on this workflow at a later time with more refined approaches. So this was phase one. Phase one really first focused on identifying what could we use at that time. And like I said, there wasn't a lot. We had Minimap 2, that was there, that wasn't a problem, but that was pretty much the extent of it. There were some quality and control tools that we could use, sure, but we had to think about scalability. So beyond, kind of scoping, I guess, the field, we actually started having to develop some of these methods ourselves until we kind of narrow down what we will do in the future. So barcode detection, right? Even quantification, we actually created our own versions of that for this initial workflow because we had no other options. And again, as I said, the goal of this first phase was to provide a pilot workflow so that UAB investigators could at least run their preliminary data. If available, we helped an individual with their troubleshooting of their original analysis. And this was also important in pre-NF core days. At this time, we weren't thinking about putting this in NF core. We wanted to, but we wanted to see where the field was going to go first. And one really challenging aspect of this, which I actually didn't realize until I started making this slide this past week, was, I had to think back about this, is that we did all of this without any robust test data. That was very difficult because, again, all the data available were the data that were paired with short read data, and that was of very low quality. So it was hard for us to validate the analysis. But we went ahead and we moved forward with what we could, and we completed phase one. And that's when phase two started. This was around 2022, and that's when some methods started to become available. So most notably of that particular year was Blaze, not only because that was something that was necessary for pre-processing long-read data, but also because the Blaze paper was one that actually released their data. So they did release a couple of great iron neurons as well as at least one Promethean sample of about 130 million reads. So we at least had some more solid data to be able to test workflows, test methods. So that was definitely a point in time that we're like, okay, we can actually start thinking more seriously about this and really testing these methods. Now, at that time, we then saw some groups, including Oxford. I think at that time was primarily Oxford and Nanopore, not so many of any other third-party groups, put some effort in trying to develop their own workflows. But these were still early days. So here at UAB or at UBDS, the road was still unclear of how this would look like in the next few years. And again, we really just wanted to support grant applications. So that's where we decided to say, let's just go ahead and move forward with implementing our version so that we could be community members here at UAB. But at this moment, things changed a little bit on the development side. We had already kind of created some barcode detection tools, methods like that. But because we saw the rise of published peer-reviewed methods, we decided to go ahead and make scnanoseq as a workflow that would standardize secondary analysis of long-read data by using, ideally, peer-reviewed approaches. We still, to this day, have some custom coding where needed, like, again, things like tagging barcodes, barcode extraction. But our hope was to actually use NF core RNA-seq has a model, if you will, right, where we're really just using and implementing as much as possible methods that have been peer reviewed, that will be maintained in the long run, so on and so forth. So we started to kind of like put these methods in place in this particular workflow, and ideally like favor that in the long term. We also wanted to make this workflow QC heavy, because that was something that we, at the time, right, 2022, workflows just didn't provide. And we did work with some investigators that had QC issues where we needed to have that quality and control metrics to be able to help them troubleshoot their actual library preps. So even things like trimming the data. I remember that Oxford's pipeline didn't have that. They deemed it unnecessary to trim based on quality, but I disagreed with that heavily. I think eventually they added that in to today's version. And I kind of consider this point in time the time where NF-core scnanoseq was actually truly born. And as a response to that, we actually officially submitted this workflow as a project to NF-core, which in 2022 included a very lengthy Slack message to their Slack channel that's called New Pipelines. And that was just the process. You always want to submit things to NF-core first, because they try not to have duplicated efforts across different pipelines. So we went in depth, we included a preliminary version of the method diagram, and then we got it approved to be able to actually develop this as part of the NF-core community. Today's process is a little bit different. I think you actually submit an issue in GitHub, and so you don't want to actually post in this channel anymore. But this is part of like the continued practice of NF-core to improve their own steps as well. So with that being said, right, we were like, okay, let's go ahead and start standardizing this. But it was easier said than done. And this is true. I've experienced this now multiple times. Right now, we're encountering this with spatial technologies. When you're working with a novel technology, which at this time it was, right, long read single cell, it's not easy to standardize analysis because the methods are so new. I could create a bulk RNA-Seq pipeline in a couple of days now from scratch if I wanted, because those methods are very, we know what we're supposed to do there. But when you don't know, you have to very cautiously think about this, because ultimately you want the biology to be right. But it's not easy to tell if the biology is right when the methods are not tested properly. And I'll give you some specific examples, but here are some highlights of, I would say, effort-heavy tasks. So that included tasks such as benchmarking and testing methods as they were released. It takes time to benchmark methods. And we really wanted to ask the question, do they actually work in the context of the biology? And even if they do, what are the limitations? So we wanted to really be very clear about that. So in addition to that, we also wanted to evaluate if there were any bugs. And there were a couple of instances where we had to fix that ourselves. And then scaling the analysis. This is definitely a key challenge for those of you who have not worked with single cell long read data. The data size is very different from short read data. We're dealing with way more data here. So this is actually, I would still consider it challenging in some methods that we use today. So we have, for instance, isoquant. We have continued discussions with the isoquant authors because that is a limitation of that tool, but they're actually making some good progress. But in response to that, in our workflow, that was a big reason why we favor also adding ORFISH, because ORFISH can do what isoquant does in terms of quantification at a much faster rate. Now, with that being said, there are still some trade-offs in there because isoquant provides a lot of different flexibilities. And then again, our vision here was to steadily make progress in this workflow so that peer review methods could be added to this. And one last thing that about this idea, right? Why did we want to add Blaze? Why did we want to add these peer review methods? In my view, a big reason for that was because we wanted to provide to the users flexibility in their analysis, because Nextflow is very modular. So if you have a process in Nextflow that uses any sort of tool that, of course, is going to be a command line based tools, you can actually access any of their parameters, even if we've not exposed that for you directly. We do try to provide to the users a set of sensible parameters and a set of parameters that you can easily customize, but it is very hard for us as workflow developers to account for every single use case and need out there. So one thing that you can do in Nextflow is that you can write a custom configuration file that overwrites what we wrote without changing the source code and enable whatever parameter you want in that particular step. Now, most of the time that will work, there may be a chance that maybe it will produce an unexpected output file where we do actually need to make some modifications to the source code, but most of the time that will work. And if it doesn't work, let us know, because then it's just a matter of us making typically small modifications. So we saw that as a way to enable flexibility, right? Enable flexibility where we're not just working with a set of custom tools or code that's going to be limited to only doing one thing or another. And isoQuaint actually is quite good at this. You can do a lot with isoQuaint, even more than what we provide out of the box. Okay, so in terms of challenges then, so what were some examples of key challenges? There were a lot, but when I went back in time to kind of, provide some slides for this presentation, I picked some examples that were like, we got receipts of this all over the internet. So I'm like, let me go back and think about what were some challenges that came with standardizing, quantifying, benchmarking. So I'm giving 3 examples, only 3 because of time, but there's more. And this is one tool, this is called Bamboo, for those of you who don't know, that enables you to also quantify isoforms. It is a great method, but it is only really more optimized for bulk RNA-seq. They just now started thinking about single cell. I was actually revisiting some of our old discussions, but I need to actually look into the details of that because it doesn't look like it's in an official release version just yet. But when we were first developing this was from an issue that we opened that got changed to a discussion later on in 2022, we were saying, hey, is there a way to implement Bamboo in single cell mode, right? And the authors replying saying, we're interested, blah, 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 right? The usual, which is great, but there was no concise plan for that just yet. But they did provide us a pathway for us to test and see if it would work. So we actually spent a lot of time, taking their method, testing this in our single cell data to see if we could get robust results and to see if we could get scalable results as well. And to make the long story short, we were not able to. It was a clear limitation of the tool, but instead of keeping that information to ourselves, we went ahead and we replied to the authors with everything that they needed to be able to do more work on this. So we provided the source code, we provided the actually even raw data from the Blaze publication so that they could easily, reproduce what we've done. Because we believed in this, in being able to be community members in these open source repositories, because maintaining a published tool is easier said than done. It's a lot of work. And if you want something to be successful, you should actively participate on that. But at the time, this is all we could do with Bamboo. So this is one method that we had to scratch. I'm still hopeful that we can add this in the near future. And it does sound like that this will be a reality in scnanoseq sooner rather than later, based on what I've seen. Here's another example, Blaze. So of course, Blaze is a method that we do have in the workflow. With that being said, even though it was a great method, it didn't come without its own set of issues that we had to fix. I have 2 examples here. So this particular one in the middle was an issue linked to reproducibility. So this is an example where the tool itself, the Blaze tool, did not provide a conda recipe or a container, and NF-core requires that. So even though this is not something that takes a whole lot of time, typically as a tool developer, it's best if you do it yourself, because we were at a position where we had to just create that for them and actually keep maintaining that. And if you actually look through that thread, people will actually say, hey, can the developers actually provide this, because we're having a bunch of people reinvent the wheel on the same issue. And over time, this adds up, right? This is not something that takes 5 minutes, still can take a couple hours for you to test. So if we're having to do this across multiple tools, it can become a non-trivial effort. But on the far right, that was actually an example of a bigger change that we made for Blaze, where Austin himself actually did make some modifications to the source code of Blaze to be able to process 5' data. So for the 10x technologies, again, you can sequence it with the 3' or the 5' method, but the 5' method was not available in Blaze. So he was actually the one that made that change himself to the source code and submitted a PR. That PR was accepted, and it's now part of Blaze. So, I mean, these 2 examples right here, I wanted to highlight this because these are 2 examples that it's a good example of like hidden effort, right? Of a lot of work that happens behind the scenes where it is many times not clear to individuals that that's what it takes. But that's the truth of it. If you do want to actually do these types of developments, you're going to want to also contribute back. And then one last example is isoquant. Again, I really actually like this method a lot. It's a very powerful approach. There's a lot of flexibility in there, but we definitely face a consistent issue with isoquant in the context of performance. Again, longer data is not trivial. It's very large, but you will see actually I've highlighted 3 issues in here where there's a clear trend that isoquant does suffer a bit in the context of scalability. And these are issues that we've actually actively participated in. They're not opened by us, they're opened by other users of isoquant, but we've actually gone ahead and participated quite a lot in these. And we've also done our own benchmark for every single tool that we do implement in this workflow. And just to quickly highlight in this benchmark, I mean, this is good because it clearly provided us a clear view of what were the limitations of some of these methods. So right at the top, you can see that the method that actually takes the longest to run on an individual sample that has 130 million reads is actually FastQC. I'm not worried about this though, because we also provide alternatives. We provide Nanoplot, we provide TulaQC, which run in about 2 hours. It's not the fastest thing. It would be great if everything ran in 5 minutes, but I'm not worried about 2 hours. So if you don't want to run FastQC, disable it. You don't need to. The pipeline provides that flexibility. Tag barcodes is something that we've actually parallelized, but it does tell us, this data indicates that we can parallelize that further. But really out in this, when you really kind of look at this picture, the one sticking point to me is actually isoquant still, right? Because it may take like about 4 hours in this particular benchmark, but this is with about 25 CPUs. That's not trivial, and it takes a lot of memory. If anything, I'm worried about both things, the CPUs, but also the memory, right? Not everybody has access to that much memory. And even if you're in Chiha, you're competing against yourself, so to speak, right? Because if you're submitting like, I don't know, 20, 30 samples, that's a lot of memory. So it was very clear that this was a gap in this particular method. And that is why we've actively communicated with the authors the need for that improvement. And it does seem like they are actually, they have done some milestones towards that. We're actually going to be looking and revisiting this very soon. But as an alternative to isoquant, we actually then provided ORFISH. ORFISH, which also quantifies isoforms, does that job in a matter of 1 hour and with far less CPUs. There's only 2 CPUs that we're using it in here. And I will note, though, that even though we highlighted the methods that we are focusing in the pipeline, we did a lot of work under the hood to also optimize this. So isoquant was also parallelized within scnanoseq. So we actually do have custom code in there that splits the files per chromosome instead of like looking at everything. And that does help with the speed, but it still suffers. So that's not sufficient parallelization. So that's why, we do go back and forth. But again, it's an excellent example of what I would call as hidden effort in computational biology. So the key home, right, to standardizing data science, especially when you're working with novel methods, is that it's far from trivial. It takes a good amount of time to really understand these methods well. It takes a lot of time to really compile and improve upon them. But I do think everybody should also try to give it back, right? If you've actually improved a method, send a PR to the authors, right? Don't let other people do the work for you. What is also another factor in the context of effort here for any NF core pipeline is the fact that this is not an ad hoc analysis. I'm not against ad hoc analysis, which is when you're kind of quickly writing a script to just get a quick job done. That can be great for prototyping, but that is far from matching the requirements for production level code, right? Production level code needs to meet reproducibility, reliability, it needs to be standardized. So a quick Python or Jupyter notebook that is not being reviewed, that's not being that's not parameterized, or RStudio, that's fine in most cases, but not for something that's going to be at a production level code. So when you do make your resource available for others to use, you want to make sure that they can implement it efficiently, and it does take time. So this is why I think infrastructure support for these initiatives are important. And I hope that we've actually, through this effort, shown to NF Core and other members that UAB has that capacity here. to pull and lead these initiatives. So that's what I wanted to kind of talk about in the context of the history of scnanoseq. And in terms of like, again, kind of like the final stage of this was to actually release it. Again, this was released in October. We're currently at version 1.2.1. But importantly, beyond benchmarking, beyond testing, we also validated, right? This is another component. Even though a tool may take a while to run, the number one thing for it is that it does the right job on the biology, right? Because I don't want a quick tool that's going to do not so good. So I wanted to very carefully evaluate these methods against what I call ground truth data, right? That I can clearly see, am I actually capturing the proper single cells? Can I quantify that? So we actually spent a good chunk of time validating all these methods against ground truth data, and we did publish this just a few months ago. So what is the roadmap to the future? Again, I would love to be able to devote my full-time job to scnanoseq, but I can't. So we have to be strategic about that. And one thing that we can do here is to participate in the NF Core hackathons, where we can devote those days for the maintenance and the development or the further refinement of scnanoseq. So this is the first time we're doing this with NF Core. We've submitted a couple of issues or PRs that we want to review. We may not be able to get through all of this because it's only 3 days, but if we can make some progress, it's better than nothing, right? So this is actually now as part of one of the projects on the NF Core, under the NF Core Hackathon. Anybody is welcome to join. If nobody joins, Austin and I will still do the work. That doesn't matter. But I am hoping to at least actually get contribution from NVIDIA. Actually, NVIDIA was here a couple of weeks ago, and Ben said, hey, we're going to be part of the NF Core Hackathon. So we actually added this one item in here, in addition to a couple of other things that we can do, which is to implement GPUs where possible. Now, not every single tool that we use is GPU enabled, but some are, like Minimap. And Nextflow last year, I want to say, or Sequera, which is the company that maintains Nextflow, worked very closely with NVIDIA to ensure that GPUs could now be implemented in Nextflow workflows. So if NVIDIA joins us, we're going to make that a priority. If NVIDIA does not join us, we're going to make everything else here a priority, although we would still want to look into GPUs in the near future as well. But beyond the NF Core Hackathon, again, ORFISH, I love ORFISH because it's very fast, but really I would love to have a standardized approach to also summarize that data into gene level matrices, just to make life easier for the users. And I already actually started this discussion with Dr. Michael Love, who is the creator, developer of DSeq2, TX-IMETA, TX-Import, because that's something that those tools should be able to support it. But again, if we cannot go down that direction, I do have a few ideas as well on how to properly summarize this data, but we have to very cautiously validate it as well. And for isoQuint, continue discussions on scalability. We'll continue to do that. Matter of fact, one more item that I'll add for the hackathon is just to upgrade it to the latest version, just to see if their parallelization has actually improved. And hopefully that will improve in the near future too. And then external contributors. We've had external contributors to scnanoseq. This is a couple of examples. This PR, which is actually a PR that we do want to focus on the hackathon if the GPU plan does not go as planned, I should say. We want to implement this PR because what this PR is doing is that it's helping with file compression. Some methods are not don't work well with compressed data, so we do have to uncompress it as part of it. And someone actually made some suggestions on some steps in the workflow where we could swap a few things, which are new tools that have been released, as well as provide additional support for file compression. Because again, this will be just storage. And again, long-read data takes a lot of storage. And then the second PR is something that will likely be coming in later down the line, but as someone is actually working with us to add this, is to add single-cell DNA processing support. So if you want to do both RNA or DNA, that may be something that is an scnanoseq in the near future. So we're heavily invested into this one, and Luke, who is the developer for this particular PR, continues to work on that quite often as well. So I am going to stop there because we are with 3 minutes, but just wanted to emphasize this last thing, Nextflow training. So what if you do want to contribute? What if you do want to be part of NF Core or submit a PR, but you don't know Nextflow, right? take a look at Sequoia's Nextflow training page. This is something that they have heavily invested for the past couple years. So they have introduction to Nextflow, introduction to NF Core. They have a few domain areas, and we will actually be bringing these trainings to UAB as workshops as well, hopefully sometime this year. But check it out if you're a good self-learner. Everything that you need to actually go through this material is already online. And even the environment itself, you can actually launch everything that you need, like in the GitHub space, to be able to learn it. So definitely be aware of this opportunity. And we will certainly be bringing that on as a workshop if you prefer that hands-on component. That's it. Yep.
Great talk. It added to my theories and stuff. So I have just one theory about when the data is generated from different platforms, is community doing any effort? Because if we do the secondary analysis, some side of clustering between different data sets from different resources generated at different places, how we are going to deal with this? Like, this is for Oxford Nanopore. What if I'm interested in multiple platform generated data and want to put them together in some sort of clustering or something?
Yeah, so you're talking about like, what if you're gonna do Oxford Impact Bio? Okay, yeah, so that's a great question. Right now, at least for the roadmap of scnanoseq, we don't have PacBio. Let me talk over here. We don't have PacBio on the roadmap, but it is something on my mind. That's what I can tell. A lot of these tools, though, they do support PacBio. So I do think that with time, it wouldn't be as much effort to actually enable PacBio data. So I'm kind of waiting a little bit to see, can we just kind of let the tools do some of the heavy lifting for us and then simply enable that type of data in the workflow or not? But if somebody does have single cell data that's PacBio and they want to collaborate with us here, absolutely. If we have an active collaboration, I'll be looking at it right away. But if there isn't an active collaboration, then it becomes a little bit more challenging on the logistical side. But again, like I said, I don't think that the amount of work now is the same as it was a couple of years ago because we've implemented peer review methods. Those methods are doing some of that work for us. So if IsoQuint lets you do it, which I know it does, if Blaze lets you do it, then it's just a matter of us maybe just processing it as normal or making minor modifications. I do think we need to make some modifications to the barcodes, but if that's the only thing we need to do, then it wouldn't be too bad, right? So yeah, so right now it wouldn't be a priority, but if somebody does want to collaborate for us, we can make a priority. And regardless, I do think it can be part of it at some point in the future. But that's a good question, because ideally you do want to process it with the same workflow. I agree, to avoid any sort of issues from secondary analysis. Any other questions? Oh, Dev? Sorry, Arash?
I was thinking about, you have different QC reports of the HTML file from each step. Have you any plans for exporting JSON or JSON-L files for the agentic systems?
Yeah, that's a good point.
Because I figured out that you can just modify some parameters, but not necessarily change the whole like toolkit using for that purpose. But if it's gonna be a loop of entry point of some JSON-L files, the agent decides any changes in that parameter and then comes back with a specific input format.
Yeah, no, that's a good question. I have not thought about that before, but that might be a good one. Now, I will say some of the HTML files that are produced, a lot of the data is repeated in the multi-QC report. So that's why we've not overly worried about that. But there are some tools that don't. Specifically, the Tula QC tool is one. Those QCs do not, I guess multi-QC doesn't have a module for that yet. But the bottom line is, yeah, outside of Tula QC, everything else should be compiled in multi-QC, but I agree that that is a great idea. I might think about that. Yeah.
Yeah. So building on what he said, obviously I know Sakura is heavily involved in sort of the Nextflow, NVIDIA, and those communities. Like, have you guys discussed maybe any collaborations with that in the pipeline or other?
Yes, so with NVIDIA for sure, when Ben was here, and that's why we actually decided to participate in a hackathon. My hope is that they can join us and help us with at least the GPU implementations, right? Because there's only so much time, and if people can actually help us grow this, that's best. So my hope is yes, with NVIDIA. And then Sakura, Sakara is awesome. I really like them, but they also have limited effort in how much they can do for each of their 87 workflows, I guess. So this is really more on the NF core side. Sakara provides great support for like things like hackathons. If we want to come in and actually have a bigger group, they'll support food, they'll allow us to get food, things like that. But I do think that it's very difficult for them logistically to work towards the every single workflow. Now, there are some workflows that they actively participate in, like RNA-Seq is a good example, right? And even Phil is involved with many workflows. Of course, he's participated in ours here and there, but I just think that's a lot of work for them. We'll see though. I will be going to the next flow summit in Boston, so I do chat with them. And then Sakara AI also is something that they have created that can help us here, and we're already started to leverage that as well. Now, that actually is probably the best way for them to provide support for such a large number of users is actually through Sakara AI. Yeah. Yeah, Nev.
Big update pipeline.
Yeah.
Just wanted to know that, so through the pipeline, is there an option, because I know from isoquant, we can also build, like transcript models.
Yeah.
But there are different tools like Squandy that does, it's a, like industry,
yes. Yeah, I'm aware of them.
For making these models, like transcript PDF models.
Yeah.
Is there a way you can like integrate that? Or is there a plan to do that or something like that?
Now, let me ask you one thing, because I know you've used isoquant separately. Does isoquant, because off the top of my mind, I can't remember, does isoquant import allow you to enable that on its own? I can't recall.
Some modifications.
Yeah, because I remember we talked about some modifications.
Then it doesn't quite accurately give me different
Okay.
categories of the
Okay.
So
if you want, so are you asking if we're planning on adding Squanty on its own? Okay.
Someone gives a Squanty like models, but then it's not just as.
So that's where community information is good, because I thought that was working since it did work when we talked, like it ran. But if you think it's not accurate, submit an issue. That's always going to be the best thing, because we need information. It's hard for us to test every single parameter option for every single tool. So if you think, hey, the Squanty quantification is not as accurate, can we go ahead and add this separately? At least it'll be on our radar, but it has to be in the GitHub. So add information if you think like, and ideally provide us something that we can also test, because how will we test, or if you want to be the person testing, right? Because we need to be able to ensure that things are working well. But yeah, submit an issue with that information, and then we can kind of at least put in a roadmap, because if it's not in GitHub, it's going to be floating in someone's cloud and it's never going to happen. So, but thank you. That's good. That's good information. Any other question?
Yeah. Do you think there is a chance of adding machine learning to the pipeline, which will mainly use for classifying the isoform classification or the read errors, mainly for differentiating the slice variants from the sequence errors?
Yeah, no, that's a good point. That probably would be something that we can do. Again, it has to be after quantification, because you need to have that in there, but I don't see why not. Now, and I think part of it goes with Dev's question, like we need to be able to just ensure that every single information that we're trying to quantify at the isoform level is present first. But yes, I think that that could be a possibility in the future too. It's definitely not in the immediate roadmap, because I'm really trying to focus on scalability, but I don't see why not, right? I'm actually thinking about machine learning for a different project to kind of help us classify some, prioritize some spatial transcriptomics data. So absolutely, I think that could be something. And if you have any ideas, happy to chat to you.
Yeah.
Okay. Yeah. Any other question? Okay.
Thank you, Lara.
Yeah.